Right. So this last section, I'm going to be telling you about sort of two other types of applications, which are sort of not directly linked to the expression matrix or the count matrix, but they're nevertheless sort of very useful, and it's important to know about them these days. So one of the consequences of this sort of a huge improvement of single cell technologies, coupled with a sort of continued reduction in sequencing costs, is that people are sort of dreaming up and coming up with more applications than they did before. So one of them is sort of really more human genetics inspired. So we can now start thinking about human sort of variation across the genotypes. So the idea is that you would sort of profile the same cell type from lots of different donors, and then if you also have their genotype, you'll be able to sort of infer which variants are sort of responsible for differences in expression levels across these different donors. So we can really start to identify these expression QTLs. And this will, of course, be very important from a sort of the basic biology point of view in understanding sort of how non-coding variants can impact expression levels, but also sort of in the bigger scheme of things, it will hopefully sort of take us further along the way of sort of personalized medicine for clinical applications. So the first sort of study that looked at QTLs in the single cell data set and a sort of meaningful level was published in 2018 from the Franke group. So they looked at about 25,000 cells from 45 different donors. So they found, I think it was 379 different cis-eQTLs. Most of these, I think about 330 or so of them, were also found in a sort of equivalent bulk RNA-seq sample. So most of the so you don't find a whole lot new, but there are some interesting features that you gain from the sort of single cell perspective. So one of those is sort of shown here. So with this variant here. So what you can see here is that you have basically different trends with sort of here you have in the CD4 T cells, you have higher expression levels if you're homozygous for C than if you're homozygous for A, whereas for the NK cells, you have the opposite trend. So what this means is that in the bulk RNA-seq, these two things essentially cancel each other out, so you can't see the effect. But in the single cell RNA-seq data, you see that you have these cell type specific QTLs, which is sort of what we were hoping to find. So again, sort of going back to the more sort of pre-processing stage of this, one challenge that you have here is that if you try to if you sequence each donor separately, then you'll have the problem of batch effects. So, but there is, of course, a way to get around that, and that is if you just pool all of your cells together, and then sequence them, then you don't have the problem of batch effects. And then we can use information about the different variants from SNPs in these cells to sort of demultiplex the cells later, and so basically identify which donor each of the cells came from. So there's major advantages that would get rid of batch effects, but it is also importantly from a cost point of view. So it allows us to sort of run more cells per sample, and which will sort of reduce the overall costs. So the first method to deal with this is called Demuxlet, and it requires, in addition to your single cell RNA-seq data set, it also requires genotype profiles for each of these each of the donors that you have in your sample. So one, you might think that it could be problematic if you have this type of droplet-based data where you only get sort of primarily the, say, the 3' or the 5' end of each gene. But it turns out that the authors here did some analysis, and they were able to show that if you have, say, 64 donors, and in that case, you only need to have sort of 50 informative SNPs in order to be able to desegregate the cells with a high accuracy. So this is indeed sort of well suited for these types of technologies. So in the second generation of these tools, they did away with a very important step here, which is having a known reference. So one such example method is called Supercell. So in Supercell, you only require single-cell RNA-seq data. You don't require the genotypes. And so what Supercell does is that it starts from the original reads, and it remaps the reads using Minimap2 and using sort of settings which are more favorable for being able to do this variant calling. Then it does the variant calling, and then it builds up this cell by variant matrix, which is then uses for clustering. And one advantage of this clustering is that it gives us sort of two pieces of information sort of falls out more or less for free, which is very useful. So one is sort of doublet detection, and it's important here to realize that there are sort of different types of doublets. So one is referred to as sort of genetic doublets. So that's when you have two cells from different genotypes. So those are the only ones that we can detect. If we have two cells from the same donor, then we can no longer detect them using this method. Then we have to use the type of methods that I talked about in the previous lecture. So in this space, the doublets come out very nicely because we expect them, each cell to be sort of very near a cluster. But if we have things that are sort of lining up in between clusters, they are very clearly doublets. The other aspect that comes out sort of for free is an estimate of the amount of ambient RNA. And the reason for that is because we know that each allele frequency should be sort of very close to either 0, 0.5, or 1.0. So if we observe sort of reads or alleles that have sort of deviate significantly from those ratios, then we can be pretty sure that that is due to ambient RNA. So transcripts having sort of bled into every droplet from outside. And so the method can actually produce what I believe is a very good estimate for the amount of ambient RNA in your sample, which is quite useful. So the second type of method that I want to talk about here is something called CITE-seq. And what CITE-seq does is that it allows you to measure not just the transcriptome, but also some levels of some proteins. So it does this in a very clever way, is that you take antibodies that are sort of specific to certain the proteins that you're interested in. So you typically have a sort of relatively small or a panel of antibodies that you can look at. And then each of those antibodies is tagged with a nucleotide sequence. And so then after we've done our sort of droplet encapsulation, we can sort of release the tags from the antibody, and then we can sort of sequence those together with all the RNA, and that can give us sort of a good estimate of the protein levels that were found in these cells. So this is, of course, there are sort of limitations. So we need to have a good antibody, and we can only look at proteins that are on the cell surface. But even with these restrictions in mind, this is very, very useful. So one thing that it allows us to do is to sort of use a sort of single RNA-seq sort of in a way that we would sort of use flow cytometry. And as you can sort of see here from this figure from their paper, this seems to work really, really nicely. And one main advantage here is that protein levels tend to be manyfold higher than RNA levels. So for the RNA-seq data, we have all these issues with high sparsity and dropouts. But for the protein data, this is typically not the case, and it tends to be sort of look very nice and Gaussian. So this is a very nice feature to have. The other thing that this has been built on is to develop this method called cell hashing, and the idea here is that you can use this for doing demultiplexing. So you can basically add these antibodies with these specific to each of your samples. You add the antibodies with the specific tags, and then you can do the sort of demultiplexing later on. So that again will sort of allow you to get overcome batch effects and possibly also save some money when you're doing your experiment. This method has been expanded, so they have CITE where they also incorporate looking at sort of VDJ sequences and gRNA. So you can combine this single-cell RNA-seq with CRISPR perturbations and do the readouts from the same cells of multiple things. So that's quite handy. So yeah, so this really takes me sort of to the end of this roadmap here. And yeah, I think we've sort of covered most of the major steps here and some of the tools that are available for doing this. What's really important to mention is that this is a very fast-moving field. So people are sort of constantly publishing new methods that sort of try to do better for these different steps that I've been discussing. But perhaps more excitingly is that people are also sort of inventing entirely new things that you can do with this data. So for example, 3 years ago, we wouldn't have been talking about RNA velocity or clustering by genotype, but now these are sort of, I would say, sort of quite standard things as part of the sort of standard processing pipeline. So I would expect there to be sort of more development like this in the coming years. So finally, what I should mention is that many of you have already talked some about SURAT, and many of you may have already have heard about it. So this is probably the most widely used and popular method in the whole field. So it's basically a toolkit that sort of will do sort of pretty much most or all of the steps that I've told you about before here. There are methods in SURAT for doing this. So I think SURAT is developed and maintained by the CITE lab at NYU, and they do a really nice job of sort of, they put together really good vignettes and documentations, and they maintain it actively. So I think now they're at version 4.0. So this is a very good package to use if you're going to sort of start somewhere. The one drawback, depending on sort of how you feel about your programming languages, is that this is built in R. Which I find a bit of an acquired taste. So one alternative is to use STANDPIE instead, which was developed by the TICE lab, and they also have sort of very nice documentation and sort of actively maintaining this, and this does pretty much the same thing as SURAT, but it's based on Python. And I think by most benchmarks, it would be more efficient, faster, and less memory intensive, because that tends to be happening when you compare R to Python. So what I should mention now is that there are sort of when I first started, pretty much everything was done in R, but over the last few years, there has really been a sort of trend that more and more things are being done in Python, and I think that is sort of set to continue. Now, of course, the good news is that there are also sort of interpreters, so it is possible to sort of call R code from within Python and vice versa. So just because your favorite package is not written in the language that you want, you don't have to switch your entire pipeline. Should be good news. So with that, I will just sort of mention there are the sort of what I think are the really hot topics in this field. And again, given how fast the field is moving, you'll probably be hearing about them next year already. So this is sort of multi-omics. I touched a bit on that, but there are sort of other types of omes that people will be comparing. And I think in particular, you'll see a lot of sort of RNA plus ATAC combined. There are already several protocols out for that, and 10x recently released a kit for that, which will mean that it will have much greater uptake. And then there's sort of spatial methods where it basically comes down to image analysis, right? Where you sort of look at a section of a tissue, and then you can put it into spatial context where each cell that expresses varied transcripts are found. So I think with that, this is the last slide. Much of this is based on some material that me and my group put together. You have the link over there. Fortunately, it's a little bit outdated, but you'll still find the sort of the overall framework should be okay. There are a lot of exercises in there, so if you want to sort of try out yourself to learn how to do these things, and with answers being available, you can go there. And then I also put a link to a very nice a review which sort of goes over these steps and tells you what they think are the best approaches to use. So with that, I will stop sharing, and I will stop the recording.